ladies and gentlemen, and welcome to lecture number 4 on cell membranes. This goes with book chapter number 5, lab number 3, quiz number 2, and is also featured on essay number 1. Sorry about that, knocked something over. Okay, now I've been very nice to you guys this first unit, and I've been telling you what goes with what. I will not be doing that for the rest of the course. You'll need to use the Guide to Biology 101 to figure it out for yourself. So anyway, cell membranes, that's the topic for today. Cell membranes are mainly made of two things. First off, our friend, the phospholipid with the phosphate head and the lipid tail. Also, there is cholesterol in the cell membrane. Now, cholesterol has this big part down below the surface of the cell, so this is the nonpolar part, and it has a small polar part that sticks up and out of the membrane. Now, cholesterol is important because it keeps the cell membrane moving properly. Remember, our phospholipid membrane is a bilayer. It has two layers of phospholipids. Heads are out, tails are in, and when you put it in fluid, you wind up with a sphere with fluid on the outside. The bilayer, the hydrophobic tails are facing each other, and then fluid on the inside. Now, one question that cell biologists asked and finally got the tools to figure out was, does the cell membrane just contain phospholipids? And they figured out a way of freezing cells and then using very delicate tools to split open the phospholipid bilayer. And they found that no, there are big proteins in here and there's holes where proteins went. So it's more than just protein, or more than just phospholipids. It's actually more of a mosaic. So the next question was, can proteins in the cell membrane move? Well, one way that scientists figured this out, and I think this is kind of neat, they actually took a human cell and a mouse cell, and they forced them to fuse into a hybrid cell. Now, mouse cells have certain proteins in the membrane, and humans have different ones. And if they were, if the proteins couldn't move around in the membrane, you would expect that the final result would have half of the dark green human proteins on one side and the light green mouse proteins on the other half. But they found that this wasn't the case. The proteins from both cells moved around after the membrane had fused. So these two experiments gave rise to the current model for cell membranes, which is the fluid mosaic model. The membrane is a mosaic. It's made up of mostly phospholipids with steroids like cholesterol, proteins, and other stuff dispersed throughout it. It also states that a membrane is fluid. The phospholipids stay in a bilayer, but they are free to move sideways, rotate, and wiggle their tails. Also, proteins in the layer can move or they can be anchored. Now, there are variations in this, so this isn't all the same. Cells of different tissues and different species have different molecules on their surface. This is why we can't just harvest pig organs for transplant in humans, because even though our organs are about the same shape and size, your body would say, "Holy cow, that's a pig heart," and start attacking it, due to the different molecules on the surface. Some proteins are anchored into the plasma membrane by protein filaments from the cytoskeleton, so some proteins do not move. Also, if you have a tight junction between the cells, like along the wall of your stomach, protein movement is restricted because the cell membranes are so close together. Now let's talk for a moment about archaeans. Remember, this is the most primitive domain of life. The phospholipids of archaeans contain reactive chains instead of fatty acids. So instead of having each phospholipid have two little fatty acid tails, it has a different setup. This causes the adjacent phospholipids to form covalent or very tight bonds. So this means that the archaeans are basically wearing their own armor. The phospholipid bilayer is very protective with these guys. And this may help archaeans live in harsh environments, such as the Grand Prismatic Spring at Yellowstone National Park and the stomach of a cow. There are different types of membrane proteins found in humans and in all creatures. These are classified by where they're hanging on and how long they hang on. So integral proteins are permanently attached to the lipid bilayer. They're not going to leave anytime soon. We also refer to a part of the protein that's anchored inside the cell membrane as the transmembrane domain. This is a part of the protein that's hydrophobic, and it's in the hydrophobic part of the bilayer. Peripheral proteins are temporary things. These attach to one of the bilayer's surfaces. Adhesion proteins are proteins like this one here on the outside of cells, and they help cells attach to each other and to the extracellular matrix. They also help cells attach to each other, and they bind glycoproteins, which is a mixture of sugar and proteins, to the surface of the cell that helps with immunity. Enzymes are another thing you can find on the surface of the cell. These are a special type of protein that helps speed up a chemical reaction. They can be integral or peripheral, so they can be permanent or short-term. And they are here because the cell membrane is a stable surface to carry out reactions. It's like, having a table to do the reaction on. And some of these carry out tasks in photosynthesis and respiration, which we'll learn about in the next unit. Receptor proteins are very important. These bind to signaling molecules, and the binding can cause a change in cell activity like gene expression, metabolism, movement, that sort of thing. These could be integral or peripheral, so short-term or long-term, and antibodies are one of these. So like your white blood cells could have antibodies on the surface that are receptor proteins. Recognition proteins are also crucial. This is a type of protein, it's out there, and it's basically the name, rank, and serial number of the cell. It's integral, it stays there the whole time, and it allows white blood cells to recognize a cell as self or non-self. So these recognition proteins are different for different species and for different individuals. For example, if you're one of the unlucky people that need a heart transplant, when you're transplanted with a new heart, even if the heart came from another human, your immune system is going to look at it and find different recognition proteins on the surface of the heart and go, well, this isn't mine, this must be an invader, we should kill it. So that's why people that have organ transplants have to take anti-rejection drugs. Another type of protein, like these guys here, these have passages through the bilayer. They actually have these open tubes that things can go through. These are passive transporters, and they transport molecules or ions through the cell membrane. This doesn't require energy. These are here for a long time, and these are things that allow water, glucose, and that sort of thing into the cell. In the next unit, you'll be introduced to a very special enzyme called ATP synthase. That's this guy here. It is a passive transporter, and it makes ATP, or it makes energy, as hydrogen ions come in the cell. Active transporters are just like passive transporters in that they transport molecules or ions, but they have to burn energy in order to do this. An example of this are calcium pumps, and we'll talk more about this later. Now, the cell membrane is said to be selectively permeable. This means that the membrane allows some substances to enter freely and keeps other substances out of the cell. This keeps a steady supply of raw materials or good things like oxygen and sugar and that sort of thing inside the cell and takes out wastes. So how does this kind of work? Have you guys heard of concentration gradients? Well, concentration is the number of molecules or ions of a substance per unit volume of fluid. The difference in concentration between two adjacent regions is a concentration gradient. Molecules or ions move from a region of high concentration to a region of low concentration. So let's say that this membrane lets everything through. The dark dots here, the solute molecules, are going to want to go this way because there's more on this side than on that side. So they want there to be an equal number of gray dots on either side. The little white dots are water molecules. They're going to want to go this way to even out the number of water molecules. Now, this works through a process called diffusion. Diffusion is the overall movement of molecules or ions down a concentration gradient. So if you've done that little kiddie experiment where you take a drop of food coloring and put it in some water, you'll see that over time it diffuses even without you mixing it so that the whole container is one color. And a substance will follow its own concentration gradient, regardless of the gradients of other solutes in the same place. So you can add 2 colors of food coloring and they will disperse at the same rate, following their own concentration gradient. Now there are some factors that affect the rate of diffusion. The first is size. Small molecules move faster than big ones, and they can go through more membranes. Temperature. Molecules move faster at higher temperatures. Also, the steepness of a concentration gradient. If you have distilled water on one side of the membrane and water from the tap on the other side, tap water has more stuff in it than distilled water, but it doesn't have that much. Versus if you had salt water and distilled water, then you've got a big difference. Also, charge matters. Opposite charges in different regions speed up diffusion. If you have the same charge, it'll slow down because the molecules repel each other. Also, pressure helps. Molecules will move quickly from a high pressure area to a low pressure area.